discuss the cell signaling pathways because in the introduction we have discussed the hormones, so different type, peptide type, thyroid, amine nature, or steroid nature. So we are actually trying to, in this lecture, we try to do how they perform their job, because we know better endocrine hormones move through the blood and reaches at their target and perform their job. So we are actually try to understand how they are performing their job, their mechanism of action. So before that, just recall your in the intro, in which we have discussed the hormone enlisted different pituitary hormone, the hypothalamus hormone, thyroid hormone. So these were the peptide nature hormone. So and the amine nature. So we use the mnemonic VAP comma for the peptide hormone. So and then for the amine nature hormone. So vasopressin, insulin, prolactin, calcitonin, and the oxytocin, melatonin, ACTH. So they are peptide nature hormone, and they then represent the dopamine, epinephrine, norepinephrine, and thyroxine. So these are the amine nature. So and here is your some other hormones are mentioned. So steroid nature, amine nature, and peptide. So through this discussion we have understood we have three types of hormones. One are peptide nature, other are the amine nature, the third one are the steroid nature. So where they travel through the blood and reaches the target. So when they have to their target cell reaches, so this steroid and the amine nature. So because our cell membrane is the lipid layer, so steroid and amine nature, so they can cross the cell membrane. So their action is intracellular. While if you look at the other peptide nature, because their protein nature, so they cannot cross the cell membrane. So their action is extracellularly. So their receptor should be present extracellularly, and while the amine and steroid have their receptor inside the cell. Okay, so let's see. So these are here is the lipid, the steroid, and the amine nature hormone. So if you follow a picture, so here is blood vessel where hormone is moving. So they are actually move in bone form. So with their mechanism. So when they came out from blood, they cross the cell membrane. So when they cross the cell membrane, so their receptor may be present in the cytosol, may be present in the nucleus. So these can cross the cell. So if their receptor is present in the cytosol, so for steroid the receptor is present in the cell, in the cytosol, while for the amine nature or thyroid nature hormone, so their receptor is present inside the nucleus. So nuclear receptor. So both have to interact. Eventually, at the last, they have to enter into the nucleus. So at the end, when they reaches the cell, so they attach with the DNA and perform the transcription, translation, new protein synthesis. So the action of steroid and amine is new protein synthesis, transcription, translation by ribosome use of. So new protein synthesis is function of the steroid and amine nature hormone. Are you good with that? So let's come back to your other the peptide nature. So because they cannot cross the cell membrane, so their receptor is present on the cell membrane. So when they attach at the cell membrane, so they do what? They activate the second messenger. So first messenger is itself hormone. The second messenger, mean another type of hormone, is secreted inside the cell. So when by the attachment of this ligand or hormone with the receptor, so this receptor is actually associated with some catalyze or enzyme, so which secrete the another, for example, cyclic AMP or cyclic GMP, IP3 system. So any second messenger. So that second messenger do what? So they can perform intercell physiological function. Their function is actually to activate some enzyme, to deactivate some enzyme. So if you compare both of them, in this sense, we have understood they are trying to synthesize something new protein. In this sense, they are trying to actually modify them. Some protein are already present, so they try to activate them or inactivate them, so modify them by phosphorylation or dephosphorylation. Repeat with that. So now let's see, for example, here is trying to show the steroid hormone. So for example, here is the hormone, here is the receptor inside the cell. So they attach with this, or if the receptor is present for the thyroid nature, so the hormone attaches here. So eventually, but both of them have to enter into the nucleus, attach with the DNA, and to perform their job to new protein synthesis. If you look at on the other hand, what is happening? So there is receptor extracellular. So they attach with some G protein, and we are coming back to this. But so as they attaches, so they cause the activation of this G protein, which release the alpha unit, subunit, and activate something. So when they are trying to activate the second messenger, which is here cyclic AMP, which causes what phosphorylation here. So they are trying to modify the enzyme function. So already here is gene transcription occur, here is just modification is occurring. Okay, so now if you look at because we have to discuss the detail of the peptide, but if you look at the receptor of the thyroid or your steroid, so actually there are different subunits, alpha, beta, A, B, C, D, E, F. So this we are looking at the structure of the receptor where your hormone is trying to, for example, here is hormone is attached. So the important thing is the C subunit and E subunit. So this C subunit is actually attached with the DNA, because we have discussed the receptor when attached with the hormone, they are enter into the nucleus, attached with the DNA to perform protein new synthesis. So they are actually attached with these zinc motifs. So these are actually these zinc motifs attached at the DNA, and the hormone attaches with this E component, E subunit, where hormone binding the hormone attaches. So this receptor has A, B, C, D, E, F, different, but important one is C and E. E attaches with the hormone, and C attaches by the zinc motifs with the DNA. Okay, so if there at the end there is COOH and NH2 is attached. So now let's look at the once again the steroid hormone, how they are performed. So for example, here is nucleus where DNA is present. So this is cell, this is your cell membrane. So let's see. So for example, here is receptor of your steroid. So let's what is happening. So for example, this hormone comes by the blood of steroid nature or amine nature. So they will cross the cell membrane, attach it with the E component. So what will happen? Then this receptor and hormone complex will enter into the nucleus. So let's see, these are attached with the nucleus. Okay. So this part of DNA, if you look at this part, this segment which is attached with the receptor hormone complex, so we name them SRE, means steroid response element. Steroid response element. So this is segment of DNA which attached with this receptor hormone complex. So other steps are simple. So transcription will occur, then with mRNA synthesis, and the translation occur, new protein synthesis, which will perform the physiological action. Are you clear with this?
Yes.
So now looking back the comparison between the lipid soluble, you can say the steroid and the thyroid hormones and the water soluble peptide hormones. So there some comparison. Receptor inside the cell, you know better, and there is outside on the outer surface. So they're inside and outside. Just follow the picture. Intracellular action, synthesis of new protein. Their job is new protein synthesis. Well, their action is they cause the second messenger system which will modify the action. Okay, so second messenger synthesis, but just remember the insulin. So they have a different type. So they do not utilize this cyclic AMP. Their mechanism of action is tyrosine kinase. We look later at. Just remember. So if you look at the storage, so they are not stored. They synthesize and secrete and perform their job. But in this case, they are stored in the vesicle. So their synthesis occur while they synthesize, store. If you look at the peptide nature, so because they are peptide nature, so their vesicle should be lipid nature, because we cannot store the chicken in the chicken box, or the lipid soluble thing in the lipid soluble vesicle. So their vesicle should be other nature. So the vesicle in which these water soluble are stored, so they are lipid nature. Okay, so they are stored in vesicle, but the vesicle nature is lipid type. So, and if you look at, so they are actually in first they are in pro hormone, then they are pro hormone. Eventually, when they are stored in the bound form. So, but free hormone is the only active form. So they are activated when there is a stimulus come from the hypothalamus or anterior pituitary to secrete the thyroid, the peptide nature hormones or any type of. Are you clear? So the exception is thyroid hormone. Thyroid hormone can be stored. So your steroid are synthesized as needed, but the thyroid can be stored. So if you look at the transport, because they are attached to the stored form, they are attached to protein and serve as a carrier. So they through carrier they move in the blood. We have seen in the picture. So exception is adrenal androgen, where they have no carrier molecule. So but they are in dissolved in plasma, are free, unbound to perform their job. So if you look at the half-life, because it takes time to synthesize them. So because the new protein synthesis, so their half-life is longer. But because they are stored for when they have to perform their job, so short time. Their need is short time. So that's why their half-life is short. But depend upon the molecular weight. The larger molecule will have little bit higher half-life as compared to the some other one. So, but here is longer half-life depending upon the bounded protein. Are you good with that? So these are comparison points you need to understand. Now, come back to the major discussion which is really tested in the most of the exam is the G protein system. So we have discussed the basic differences between the peptide and the lipid soluble and water soluble. Now come to the major discussion of G protein. So before discussion of G protein, you need to know the phosphorylation and dephosphorylation. Phosphorylation means attachment of phosphate with some substrate. In this case, for example, here is your substrate. If you look at here, so it is phosphorylated with when the phosphate is attached. So for this we need kinase enzyme. So kinase do what? Phosphorylate some substrate. The other term we use is which will dephosphorylate is the phosphatase. So phosphatase and the kinase, two terms. Kinase to phosphorylate something. For dephosphorylation we need phosphatase. So they remove the phosphate from the substrate. Okay? Phosphorylation. So we have discussed the function of the peptide nature is to modify them. So either they phosphorylate them, either they dephosphorylate them. So for example, if you look at the biochemistry, so for glycolysis or gluconeogenesis, so different. So hormone, we will say insulin is performing this job or inhibiting or the stimulating. So they actually do nothing, just phosphorylate them or dephosphorylate them. So by kinase enzyme activity or by the phosphatase enzyme activity. So we need to actually, so this kinase is actually should be activated. So it has actually 4 components. 2 are regulated, 2 are the excitatory. So they are activated by the second messenger, because their second messenger do what? So they activate these kinases or phosphatases. So they have four subunits: two are regulatory, two are the catalytic. So to perform the job. So let's see what is happening. So for example, here is second messenger 2. When they attaches, let's see. So the second messenger attaches with this part. So what will happen next? So this part, the catalytic side, separated out from this one. So now it's have ability to perform the job. So we are actually trying to discussing the different second messenger which need to activate the kinase, different type of kinase. So they have four subunit when they are activated by the second messenger. So this second messenger is actually G protein coupled receptor type. So mean there should be a G protein, there should be a receptor, and they should be coupled by the word. So they will activate the second messenger. So there may be a cyclic MP or the AP3 calcium or the cyclic GP. We are going to look at this system later. So before that, we just look at the G protein coupled receptor structure. For example, here is the cell. So you do better their receptor are extracellularly for the peptide nature. So for example, here is receptor, this one. So they are attached with these 7 with the G protein, these 7 helical of the 7 transmembrane helical. So they are back forth. So there are 7 curling back and forth movement. So this transmembrane are polypeptide. So attaches the receptor with the G protein. You can see here. So we discuss G protein here and the coupled receptor. So they are coupled by this polypeptide, which is 7 back and forth transmembrane helical. Are you good with it? Yes. So this G protein has actually have 3 subunits: alpha, beta, and the gamma. So alpha, beta, and gamma 3 subunits. So major important is the alpha one. So alpha unit, beta unit, and the gamma. So this has the ability to activate the second messenger system. So most of the time it's attached with the GDP. So when the receptor come, the hormone come attached with the receptor. So this alpha GDP attaches with the remove the GDP and activate attaches with the GTP, alpha GTP. So we will look later what's happening. So for example, here is the hormone. So when they attaches, what will happen? So this alpha GTP will attach with the GTP now. Are you good with that? So what is happening next? So you can see it's separated from the G protein. So this alpha GTP has ability to perform the second messenger to activate the second messenger system. Okay. So this when the hormone stop, so this go back into the GDP form, inactivated, because no need to perform the second messenger, activate the second, because there is no hormone is coming. So so the factor which activate this system, we name them GEF, and the factor which inactivate them GAP. So what does mean by GEF? GEF is actually gonosine nucleotide exchange factor, which you just no need to remember the term. So just remember these are ligand. So ligand are a hormone. So they activate into convert the alpha unit GDP into alpha unit GTP. So another GAP, which is GTP is active protein. So in this case is the alpha subunit. So when there is no hormone, so this alpha unit have itself ability to come back to the GDP. So they activate itself. So when the no hormone is coming. So let's see what is. So they do what they call the hydrolysis, mean remove the phosphate and become the GDP, triphosphate into diphosphate. Okay. So now this is actually G protein system. So let's see how if you have this basic idea, you can easy with the cyclic AMP, cyclic GMP, AP3. So because this is major confusion concern. So let's see what is happening. So for example, here is protein kinase A. We need to activate them. So we need a G protein, which is, let's see, G protein, which is attached with the alpha, beta, and gamma subunit receptor is here, transmembrane is here, and for example, here is hormone attaches. So as the hormone attaches, what will happen? This will activate. So alpha GTP will separate out. So now this alpha GTP has ability to activate the second one. For example, enzyme which is attached with the cell membrane, adenine cyclase. So this will attach with this. Let's see. So it is attaches here. As they attaches, they do what? They convert the ATP into cyclic AMP. Okay. So this is activated by the alpha and GTP system. So when they activate them, so they convert the ATP into the cyclic AMP, cAMP. This is one of your second messenger. Now it's activated. Do what? It will go to the activate the protein kinase A. So why we name? Because it's kinase, because it's activated by this adenine cyclase. So we name them protein kinase A. Just later. Okay. So they will, for example, modify the different perform their physiological function. So this is cyclic AMP system. So this is actually GS system. It stimulated. GS are stimulatory. We have GS, GI, and GQ. GS is stimulatory. GQ is inhibitory, and GI is inhibitory, and GQ is actually associated with the IP3 system. So GS and GS and GA both are associated with the cyclic AMP. One will stimulate, this other will inhibit. So let's see the GA. What is happening? So you know about protein kinase A, and the receptor attached with the G protein, alpha, beta, and gamma subunit. For example, hormone come attaches here, so they will convert into alpha GTP, and they will attach with the adenyl cyclase. But in this case, because it's part of GI, so they which conversion of ATP into GTP cyclic AMP is the normal function. But in this case, they will inhibit this because this is activated by the GI. So when GI activated, they inhibit this conversion, this activation of the cyclic AMP. So they decrease the cyclic AMP level. While the GS do what? They increase the cyclic AMP level because it's stimulatory, it's inhibitory. Okay. Now come to the other one, the IP3 system, GQ system. So for example, here is protein kinase A. We need to activate them. Let's see what is happening. So here is receptor transmembrane attached with the G protein, alpha, beta, and gamma subunit. For example, here is the hormone. So hormone attaches here, activate the alpha GTP, which will activate the phosphokinase C, which is a hormone attached at the membrane. So this alpha GTP do what? So they attach with the phosphokinase C, and when they activated, so they convert PAP2 into IP3 and the DAG. So these two factors, IP3 and the DAG, when the phosphokinase C is active. Okay. So let's see these two, how they are performing there. Actually, when we discuss the IP3, this one we use the word calcium IP3. So because their function is associated with the calcium, actually endoplasmic reticulum are present inside the cell. So they affect the endoplasmic reticulum and cause the release of calcium. Let's see how they are performing their job. So for example, here is endoplasmic reticulum. So this IP3 system attaches, so the gate of the calcium opens. So calcium rushes out. Okay. So this calcium is by the IP3 system. So this calcium and this DAG cool, will activate the protein kinase C. These two one, the calcium by the AP3 system which has come out, and this DAG will activate the protein kinase C. So they are both are trying to activate the protein kinase C. Are you good with that?
Yes.
Okay. Now we have discussed all these pathways. The next problem is to remember the hormone which use the second messenger cyclic AMP, which use the AP3 system. So there are some mnemonics I have used. So just follow them, which will help you to answer a lot of questions. Before that, PK, so which you have discussed, the protein kinase. So their action is actually the serine threonine phosphorylation cause and the enzyme phosphorylation. So either they, when they phosphorylate, they become active or inactive. So by the phosphorylation, and they activate the CREB, and which will response element binding, actually it's a cyclase. So they will also gene expression and the ion channel, so they may be open or closed. Where this PK, these are actually function of your PK. When this system is activated, they do what they can do. So, and your epinephrine system, these are important points, especially for step 1 exam. So, which epinephrine and acetylcholine? So, because epinephrine is part of adrenomedullary sympathetic system, so when how they perform different job by the actually using different second messenger system. So, for example, if this epinephrine attached with the beta receptor, so they stimulate the GS system. When they attach with alpha 1, they activate the GI system, alpha 1 receptor. When they attach with the alpha 2 receptor, though they activate the GQ system. Okay, GQ, which is IP3 related. If you look at the acetylcholine, so when they attach with the M1 receptor, so they stimulate the GQ. And when they attach with the M2 receptor, they stimulate the GI second messenger system related, and M3 also the GQ system. So now look at the cyclic AMP. So for cyclic AMP, just remember flat, champ, chug. Flat, champ, chug. So these are hormones which follow the cyclic AMP. So use the second messenger as a cyclic AMP. So, and the IP3 are the goat egg. So let's see what are the hormones. So flat mean actually your flat hormone, flat PG, FSH, LH, ACTH, and your TSH. So, and champ represent actually your CRH, histamine 2. Histamine is actually 2 receptor. One is H1 and one is H2. H2 use this pathway. So, and ADH and your MSH and your PTH hormone. So, and CHUG represent actually calcitonin, HCG, glucagon, and GHRH, growth hormone, releasing hormone. So these all hormones use the cyclic AMP pathway. Okay, and let's see the IP3. So goat egg, G represent GnRH, here is GHRH, which is growth hormone. Here is GnRH, which is FSH and LH related. So, and oxytocin, ADH, here is ADH V2 receptor, but in this case V1 receptor is utilized. So, and your T represent TRH. For HEG, histamine, H1, and angiotensin 2, and the gastrin. So these are far easier way, because here is H1 receptor, histamine present here, ADH is present here, also present here. So easier way, because here is I. If you follow the picture, because I look like 1. If you I and 1, so they are both are look like same. So just follow IP3, use a V1 type of receptor. So V1 and H1 are by the IP3, while the V2 receptor and H2 are cyclic AMP. I and 1 look like same, so easier to reach out. Okay. So this is actually mnemonic which will help you to understand the to identify the which hormone use which pathway. So now come to the one clinical which is really important. So for this toxin cholera and the other is the Pertussis and whooping cough. So one is intestine related, the other is respiration. So before that, I want to recall I use a mnemonic for understanding accident example. So accident is occur due to two conditions. If you have overspeeds, you have crossed the speed limit, mean you are accelerated mode. Or accident occur if you lose the brake. If your brake is not working, you may lead to accident. So by the overspeed, if you have stimulated something more, or by you lose the inhibition. So you know better, as we have discussed cyclic AMP system. So GS is stimulatory, means speed. So if you have a more activated GS system, mean there is accident. That is here toxin cholera. And if you lose the GI system, mean you have lose the brake. So that condition is Pertussis and whooping cough. Okay, because one is the inhibitory, one is the stimulatory. Stimulatory because when you overstimulated, means cyclic AMP synthesis will be more. If you have loses the inhibition, in both cases cyclic AMP level go up, because we are stopping the inhibition. So cyclic AMP is increased. So both conditions are the cyclic AMP related. Number one concept. In both conditions cyclic AMP go up. And the second important thing is that difference is here is GS, here is GI system. And here it is in the condition happening in the intestine. This is happening in the respiratory tract. Okay.
Toxin cholera is due to increased stimulation.
Stimulation, GS system, and this is due to GI, lose the GI function. Okay. So GS, everything is mentioned. Don't worry. So GS is more stimulated. So what will happen? Cyclic AMP will more, and this will PKA will be activated more. Protein kinase is associated with. So if you look at which will activate the actually CFTR factor. So which is actually your response, PK response element. Actually this is. Okay, GI is in a function is lost or decreased. So which will also increase the cyclic AMP, and PK function will go up, and this actually cause the loss of mucus and fluid and electrolytes. So that's why they call a diarrhea condition. So because they lose different electrolytes. So if you look at here, for example, cyclic AMP is activated more. So which will phosphorylate this CFTR, because the function of we have discussed the PK function, activate the CFTR. So when they activate it, they do what? So they cause the release of a lot of electrolytes. Water, chloride, potassium. So different ions are come out of the cell. So when these cells come out of the cell, so they lead to diarrhea. Okay. So these actually toxin cholera, Pertussis, whooping cough, or black cough, which you commonly use in the village. So these are actually by this mechanism is happening. Okay. No. So we have discussed the cyclic AMP, IP3, and we are going to look at the cyclic GMP pathway. So their mechanism is same. We have to activate the kinase. So here is protein kinase G. So their receptor either present on the cell membrane or inside the cell, intracellular or at the cell. So for example, here is soluble guanylate cyclase, which is inside the cell, and other, for example, receptor which is present on the membrane, that is membrane guanylate cyclase. Both are the guanylate cyclase. One is the inside the cell, we name them the soluble. One is at the membrane, we name them the membrane guanylate cyclase. Let's see. So this is actually important in when we use a nitric oxide, which is NO, which will dilate the vascular activity or relax the blood vessel. So how they are performing job we are looking at. For example, here is your cell, here is your blood vessel through which the nitric oxide is coming. So let's see what is happening. Actually there is L-arginine in the nerve cell. So this will convert it into the NO, the nitric oxide. So this nitric oxide is actually activate this system. So, but before that, it's coming into here. So we need to enzyme which will do the conversion of L-arginine into the nitric oxide. That is the nitric oxide synthesis. But this system is actually activated by the same IP3 system. So you know better the receptor, the GQ system, by the GQ system. So and a hormone, for example, attaches here. So when they attaches, they do what? When this alpha GTP attaches with your PAP, and they do what? So they activate the IP3 conversion into and DAG. So IP3 you know better attaches with the endoplasmic reticulum and cause the opening of the channel. So when the channel open, calcium comes out. So this calcium can attach with your call modulator, which is in the muscle. You know better because this is muscle cell. So when this attaches with the calcium call modulator, actually this calcium call modulator complex can activate this nitric oxide synthase. Okay. So this nitric oxide enter into the cell for the relaxation of vascular activity. So this nitric oxide can sometimes given extracellular by the drug, so exogenously. So both if they come by this pathway or if you give the exogenously, function is to relax your vascular activity. Okay. So this drug, by the drug when they enter, so they can enter into the cell, and what will happen eventually? So this will attaches here. As they attaches here, what will happen? So they cause the GTP because GTP into the cyclic GMP. This is second messenger which we are going to actually discuss it. In that case, ATP was converting into the cyclic AMP. In this case, GTP is converting into the cGMP or cyclic GMP by activation of this soluble guanylate cyclase. And if you look at the other cell, so this will activate the protein kinase C or the activation of. So if you look at the membrane guanylate, it is actually activated by the ANP or BNP, atrial natric uretic peptide, or the your B type natric uretic peptide, which is coming from your heart, one of the hormone. So they use this pathway. Actually, when they attaches here, so they do same function. So for example, it attaches ANP or BNP attaches here. So what will happen? GTP conversion into the cyclic GMP. So this cyclic GMP activate the protein kinase C. Okay. So one of the important related this cyclic GMP function, which is suited with your vision system. So actually, when you look at the vitamin A function, or when you discuss the neuroanatomy of your vision system, so this cyclic GMP actually, so when there is dark condition, this cyclic GMP is actually active. So this cyclic GMP opens the sodium channel in the cell membrane. Sodium comes in, and they will lead to different pathway steps. So they stop the glutamate and the bipolar synapses. So we do not see in the dark. But when the light comes, light actually causes the synthesis of PDE, phosphodiesterase. So this PDE actually destroys the cyclic GMP. When no cyclic GMP, mean no opening of sodium channel. So we can visualize something. Okay. So this concept is actually discussed in the neuroanatomy and the vitamin A discussion. So just see here what is happening. For example, this is actually not a hormone, it's actually light. Just think about it. So when the light come, so they do what phosphodiesterase activity. So when this is active, so this actually cause this PDE, release of PDE. PDE do what? They destroy the cyclic GMP, which is present inside the cell. So they do what? They actually convert this cyclic GMP into the 5-GMP. So in this way, they destroy the function of cyclic GMP. So this is actually the pathway vision system important. So your cyclic AMP or AP3, they are continuously inactivated by this phosphodiesterase most of the time. But when the hormone come or their stimulation come, so their PD function stop and the cyclic AMP and cyclic GMP or IP3 activation continues. Okay. Now come to the last one, which is actually important for the nerve growth factor receptor, insulin receptor, and which is actually tyrosine kinase system. So there receptor tyrosine kinase and tyrosine kinase. So same thing, that is kinase. But for example, here is a cell membrane, here is cell membrane, cell membrane. So just remember what is happening. So this is extracellular side, this is intracellular side. For example, here is the receptor. It attaches with the transmembrane, single transmembrane. So in this case, the concept is that in last this G protein system, we have seen the kinase was present inside the cell, which was activated by the second messenger. But in this tyrokinase system, the kinase is directly attached with the transmembrane. It's not present inside. So when the hormone activates the receptor, so they will activate the kinase. Kinase do what? Phosphorylation. So, for example, here is receptor, here is transmembrane, which attaches with the tyrosine kinase. The kinase name is the tyrosine actually. So when this is activated, so they cause phosphorylation. Phosphorylation of not something else, it causes phosphorylation of itself. Okay, we name them, that's why autophosphorylation type. So let's see what is happening. So for example, here is a hormone. So this hormone attaches here. So in next step, these two, the receptor and these system will dimerize, become one, dimer mean. So let's see what is happening. So they become a dimer. When they dimer, so their cross phosphorylation, this tyrosine phosphorylate this one, this phosphorylate this one, cross phosphorylation mean. Okay, so let's see. So phosphorylation of these, phosphorylation of these. So this is actually phosphorylating this. When the hormone attaches, first step is dimerization. So when they become a dimer, so this phosphorylate this one, this phosphorylate this one. So all the tyrosine kinase will be activated and their phosphorylation will occur. So their example is nerve growth factor receptor. So they utilize this pathway. So another example which is also similar, but in this case, dimer is already made. You can see they are attached by this disulfide system. So there is already the dimer is formed. So what will happen next? So they are attached with the tyrosine, same thing. And for example, here hormone come. When the hormone come, so they will activate them. So what will happen? The phosphorylation occur, crisscross. So in this case, smaller difference, that is the dimer is already made. In this case, by the activation, this dimer was formed. Now come to the tyrosine kinase associated receptor. It is actually because a little bit different. So in this case, we have seen the phosphorylation, autophosphorylation was occurring, and here was dimer already made. In this case, actually, when these tyrosine actually activated, they cause the phosphorylation itself and also phosphorylate some other protein which is present inside the cell. So let's see what is happening. So for example, here is a receptor, here is a transmembrane JAG protein system. So when they activated by this protein, so hormone, so what will happen? For example, here, so they cause the dimerization first step. As a dimer form, what will happen? Phosphorylation, crisscross phosphorylation, you know better. So this, when this JAG phosphorylated, in the next step, what will happen? So they phosphorylate something else, that is stat. Okay, so here is, for example, stat is attached another protein which is inside the cell. So they cause the phosphorylation of this stat. So this JAG actually causing the phosphorylation of this stat system. So when this stat is phosphorylated, so they perform the physiological function by dimerization. So in first step, they two become one, and then next step they perform the physiological function. Let's see what is it. So in first step, the physiological become a dimer, then they perform inside the cell their physiological job. Last time I reviewed all these steps. In this system, we have seen as the hormone attaches with the receptor. So in first step, dimer form, and in next step, tyrosine cross phosphorylation, or this was autophosphorylation because they are phosphorylated itself, and this perform their actual physiological function. If you look at in this system, dimer was already made. As the hormone attaches, so they again phosphorylation of itself. Okay, in this system, what will happen? Hormone attaches, dimer form, JAG activated. As the JAG activated, they phosphorylate something else, that was a stat. So stat phosphorylation occur, and they become again a dimer and perform the physiological job inside the cell. Okay. So this was actually discussion. So just remember the hormone which use these pathway. So the receptor tyrosine kinase are insulin and the growth factor. So easier way to remember IGF or insulin like growth factor, platelet-derived growth factor, fibroblast growth factor, or the epidermal growth factor. So they are all growth factor. Growth factor and insulin use the receptor tyrosine kinase system, and the other one, tyrosine kinase associated receptor system, was by the cytokines, the gamma, IL-2, IL-6, and your bone marrow cell, for example, erythropoietin, and your granulocyte colony, stimulate factor, or thrombopoietin, or the other, for example, your flat PG and the prolactin and the growth hormone. So these are actually using these pathways. Okay, so this was actually our discussion of the cell signaling pathway. So we'll stop it here. So thank you very much.